Good evening to everyone. Today we are going to tell something about the Mendel's law of the animal genetics. Mendel's law of the animal genetics, and we will go ahead with the lectures as mentioned in the particular thing is concerned. Then the Gregor Mendel, who lived during the year 1822 to 1884, is a great scientist. Then he is an Austrian monk. He born in 1822 near Brunn in Australia in a poor family. Then he joined the Augustinian monastery. So John Gregor Mendel is one of the greatest scientists who is the father of the genetics. Hence, the person, especially he is presented his findings the before the National History Society of Brunn in 1965. So Gregor Mendel is one of the person who is in pioneered in John Gregor Mendel is pioneered in the animal genetics and breeding. Then he is an Austrian monk who born in 1822 near Brunn in Australia in a very poor family, of course. Then he joined Saint Augustine Monastery and was sent to University of Vienna. Then the presented his finding before the National History Society of Brunn in 1965. So he is a person who has evolved the law of the genetics or the Mendel's. So his findings were discarded due to the prevailing theories of evaluation. Later on, these theories were again discovered simultaneously by Hugo de Vries of the Holland and Karl Lorenz in Germany, then Eric von Tussermark in Austria in 1900. So later, this theory was propagated, and once again, the Mendel's laws were considered, and he was considered as father of the modern genetics. Then he actually invented why a pea plant is there and whether the male sex structures like stamen and female sex structures as the pistils, very similar to like this. Then he also found the power flower structure on pea ensured self-pollination and also well-defined male and female parts. Till that time, the male and female parts, it was not known that it is going to be caused due to the pollination than by the bees. And it was a pea nut was a single season crop and accordingly it was invented. Then emasculation and pollination is the concept. Then the earliest 1900, the flower is quite easy when they made it, especially the plant genetics was very popular. Then there was a concept that the reproduction was happening with sexually. Then in the varieties available, several characters had two contrasting forms which were easily distinguishable from each other. For example, the hypothetico, that's the deductive method applied by the Mendel, apparently unknowingly. So he proposed several theories like observations, then the observations were hypothesized, then experimental predictions were also done, then testing by experiment. Once again, the observation, ultimately it led to some theories. That's called as the Mendel's theory. Then initially, these theories were proved on the plants because it was very easy to find the reproduction, especially or the genetics, especially in the plants. Hence, the tall and dwarf plants, or tall and short plants, then axial and terminal plants were seen, and the seed structure and seed color, then pod color, then pod shape. All these were observed while crossing in between the tall and short and axial and terminal plants, and accordingly the seed color, the plant color, etc. They were being changed after the things are being considered, and hence there was a difference. So these are all the genetic experiments which were initially conducted by the Mendel. Then the it became very popular as the Mendel's law of inheritance. Then also in case of the animals, initially it was in the plants and later it was percolated into the animals also. So this was very popularly known as the Mendel's law of inheritance in animals, and overview of the Mendelian genetics principles. So we need to examine these principles in detail as such, and we need to examine what are all the inheritance factors which are present in the Mendelian theories. So but first it told law of segregation. Then what's a law of segregation? Each individual has got two alleles for each gene. That was the first sentence, one from each parent, either male or female parent. Then it was called as the alleles segregate during gamete formation. Means either it may be a sperm or egg, whatever it may be, it is called as the gamete. Then each gamete carries only one allele for each gene. Then offspring receive one allele from one parent. So restoring the pair. So this was the theory of or the law of segregation, means each individual has two alleles for each gene, one from each parent, maybe male parent or female parent. Then it is inherited from one allele to the another allele. So the gamete formation is also called as the sperm or egg cells. Then each gamete carries only one allele from each gene. Now it is said that the X and Y chromosomes. At that time, the concept was not clear, but now it is very clear that all these are due to the chromosomes and the genes. Genetically, these are all prefixed. So the offspring receive one allele from each parent, restoring the pair, pair of the allele. And the law of independent assortment was another law, means the principles of the Mendel's theory. Then genes for different traits are passed independently of one another. Means genes of different traits are passed independently of one another. Then inheritance of one trait does not influence another if genes are unlinked. Then this applies mainly to genes located on different chromosomes. So the law of independence assortment is always a assortment in which it applies mainly to the genes located on the different chromosomes. Here the pattern of the chromosome came, and this is called as the inheritance of one trait does not influence if genes are unlinked. That's why the different theories were made. And the another law is law of dominance, means an individual with two different alleles has one dominant and one recessive genes. Means an individual with two different alleles have got one dominant and another one is the recessive one. So the dominant allele determines the appearance of the organisms. Means if there are many characters, especially in a species like color, the height, the breed, the milk quality, and other thing, then one will be dominant and another will be recessive or suppressed in nature. That's what the law of dominance says. Always the dominant allele get expressed, whereas the recessive allele will not get expressed. That is the called as the dominant of the alleles. And dominant allele determines the appearance of the organism. Then recessive allele is hidden unless both alleles are recessive. That's what it says. Then the application in case of the animal. How we can apply the Mendel's law in animal genetics and breeding is it is used in the breeding to predict genetic crosses outcome. So, for example, a good coat color or the different traits, they are called as the genetic traits and used in breeding to predict genetic crosses outcome. And it explains the genetic disorders from recessive alleles. So there are many genetic disorders which may be or may not be expressed, and if they are not expressed, they are from the recessive alleles. So, independent assortment contributes to the genetic variation and evaluation. So, it is also another application, especially in case of animals, because the independent assortment contribute to genetic variation and evaluation process. So there are a lot of terminologies also there in case of this Mendelian law of the inheritance. One is the character. So character means the morphological character, the anatomical character, behavioral, then feature of an organism, maybe starting from an insect to ending up with the elephant, or maybe the biggest mammal, that is the whale. Allele different alternatives to form genes. Example, a gene for height has two alleles, one for tall and one for dwarf. Whereas the homozygous, an individual with two identical alleles means allele means a different alternative form of the gene. For example, a gene for height has two alleles, one tall and one is the dwarf. Whereas the homozygous means an individual with two identical alleles means both are the tall alleles. Whereas the heterozygous means an individual with different alleles. Whereas the phenotypic character is observable characters by the external appearance of the individual, like maybe the color, the size, form, and the structure. That's called as the phenotypic characters. For example, the somebody will be tall and the dwarf. These are all phenotype, means physically which is appearing characters. For example, the genotype is we are unable to see it by the external morphology, and hence it is the genetic composition of an organism and cannot be seen by the naked eye or the routine process by observing it. For example, the TT or tall means small TT or dwarf, and one tall, one dwarf. So like this, the terminologies are evolving. And another thing is the F1 generation. What is F1 generation? Especially in case of the animal genetics, we are studying it. It is the hybrid offspring of the parental generation. Whereas the F2 generation, the self-fertilized uh, progenesis of F1 plants or the F1 animals. So F2 means it is derived from the F1, whereas the F2 means the self-fertilized progenesis of F1 plants, especially in case of plants. Whereas the monohybrid cross, a cross between parents differing in only one trait or characters. So here the phenotypic characters, only one trait or the characters may be seen. Whereas the dihybrid cross, a cross between parents offering two traits or characters. Then test cross is a cross of F1 hybrid to recessive parent type, and back crossing is crossing F1 hybrid to one of the original parents. So this is called as the back crossing. So here most of the experiments are done on the plants by the Mendelian theory, just like the remove. What they did is they removed the stamens from the purple flower. They removed this by the scissors. Then the carpel is also known as the female. Then this is the parental generation, whereas the white flower with the stamen, male flower, they were retained. Then the second step is transfer pollen from the stamens of white flower to the carpel of the purple flower. So white flower, then purple flower. Then the pollinated carpel matured into the pods like this. Then the fourth one is the planted seeds from the pods. Then examine upbring all purple flowers were expressed instead of the white flowers are being expressed. Hence, here the purple is dominant whereas the white is recessive. So, this is the first generation offspring and also called as the F1 offspring. Whereas the Mendel's law of inheritance also did experiment on seeds, flowers, pods, and the stems in the initial stage. Then sometimes the seeds are round in structure, then sometimes wrinkled in structure, then the cotyledons were yellow, green, etc. Whereas the flowers are also sometimes white and violet, whereas the bean shapes or the pod shapes are full or constricted. And color also differs from green or yellow. Then stem, it took place as the axial pods and the flowers along with the long and also the short type of the terminal flowers. So the Mendel's law explains why this is happening because one is dominant and another one is the recessive. So the law of dominance is always it is told by the Eric Wang. Then every gene has two alleles that can code for a trait. Every allele has two gene has got two alleles that can code for a trait, and one allele is dominant, meaning it will always show or the dominant, whereas one allele is recessive, means it will be masked by the presence of the dominant allele. So tall plant here TT, it is represented as TT, whereas the dark plant it is containing t small t. Whereas if we cross these two things, then there will be a dominant and recessive. So this is the F1 will be a tall plant with the one, the capital T and small t. So this is the theory that one allele is dominant and always it is being expressed, whereas one allele is recessive and it is not expressed. Whereas the F1 is a tall plant, then F1 the tall plant, another one with the same genetic thing. And if you cross these particular uh, plants, then the you can see the pattern here, like it goes on. And there will be a completely dwarf plants with dwarf alleles. Whereas a tall plant may also have got one of the uh, dwarf. So this law was not fully accepted because that is co-dominance was the thing and incomplete dominance was another thing. Whereas the over dominance was also the third thing was there and hence it could not be accepted. Then law of segregation we have already discussed. During the formation of the gametes, the paired alleles separate randomly so that each gamete receives one allele or the other. So this is called as the law of segregation, means two alleles of a gene present in F1 do not contain each other and they separate and pass into the different gametes in their original form, producing two different types of the gametes in equal proportion. So this can also be applied to the animal genetics also. So the pair of alleles, suppose there is a dominant allele and the recessive allele, then they separate and only one allele passes from each parent onto the offspring. So, which allele is in a parent's pair is inherited is a matter of the chance. That's all. Here, nothing is dominant. And segregation of alleles occurs during the process of gamete formation, or it's also called as the meiosis, means cell division. Then these are randomly going to unite at fertilization time. So here the examples are being given, especially once again the plant. Then Mendel's experiment, he crossed tall and dark varieties of the garden peas, P. So then the P generation first, he crossed tall with that of the dwarf, and he got the F1 generation with tall and the trait with small. Whereas when the F1 is self-fertilized, means without any cross-pollination, both tall and dwarf progenies were produced in F2 generation. So whereas the 75% were tall and 25% were dark. So for example, an F2 of 1064 consisted 787 tall to the extent of 75% and 25% 277 dwarf, almost 3 is to 1 ratio. So this is the experiment which is conducted by. So when F1 plants from Mendel's experiment were crossed back to the dwarf variety, half of the progeny were tall and half were dark. The phenotype ratio observed was 1 is to 1, and this further demonstrated more conclusively by the principles of segregation. Then when similar experiment was done on the characters, then this is the result of the Mendel's result in cross between the plants differ in 1 7 character. So these are all given as the seeds with smooth versus wrinkled, then yellow versus green, gray versus white, purple versus white, axial versus terminal. So all these are very much complicated in thing. Then ultimately, the what is the ultimate end product is it is calculated with that of the different numbers of the dominant and recessive genes. So totally they have calculated how much quantity of the dominant versus recessive in every type of the phenotypic character, smooth versus wrinkled, yellow versus green, etc. Then the ratio was almost 3 is to 1 or 3.4 is to 1. And the dominant trait is always written first, usually T or the, for example, the T capital, then we are followed by the small one. So a single gene controls both the seed coat and flower color trait in the Mendel's experiment. Then law of independent assortment means this states that the member of different pairs of the alleles assort independently during the formation of the gamete. And this means that traits are transmitted to offspring without any uh, dependence on any of the alleles and independently to the one to the another. So Mendel crossed plants that differ in the genetic traits or alleles. For example, the round green seeds and wrinkle yellow seeds. And he got the F1 hybrids which were round and yellow seeds as the expected. Whereas when the F1 were self-fertilized or pollinated, 4 F2 phenotypes were observed. Then from a total of 556 seeds, so there was a distribution of 305 round green, 100 round yellow, 100 wrinkled green, and 32 wrinkled yellow. So this was a permutation and combination. So these results closely fit with a ratio of 9:3:3:1. So when Mendel crossed the F1 generation obtained with the recessive parent, 4 different phenotypes were obtained. Then it verified the ratio in which the different gametes of F1 hybrid were produced. Just like here you can see that the phenotypic characters are in the formation of the ratio of 1 is to 1 is to 1 is to 1. So the parents and the offsprings share the different phenotypic characters. So this is also depicted by this figure where in which the genetic analysis is being done, like just like the peas, especially 9:3:3:1 ratio has been depicted in this particular autograph, wherein the F1 generation is crossed, whereas F2 is self-pollinated. Then this also shows the same experiment which is produced further with the different types of the seeds, round yellow, round green, then wrinkled yellow, wrinkled green. So all these are the outputs of only two, that is round and yellow with wrinkled. So two colors. Then how it has happened means the it has caused different generation like F1 generation where these are segregated. Meiosis happened and fertilization happened and the gametes were formed. All F plants produced yellow round seeds. Then you can see the cascades of the events where the gametes they produced the ratio always ending up with 9:3:3:1 phenotypic ratio of the generation. So this was applied very early in the plant generation. Later it was evolved into the different animal breeds and also different crossbreeds, poultry, then piggery and many more fields of the area. So the Mendel was such a successful person in initial days because he studied the inheritance of only one pair of the contrasting characters at a time, only one pair. Whereas he selected P varieties that are clearly different forms of one or more character. Means he selected such characters which are exactly contrasting to each other, just like yellow and purple. As for example, the phenotypically they should be easily identifiable and differentiable. So he took such traits just like round and wrinkled seeds, yellow and green cotyledons. So he carried out his experiments with great care and the elaborateness. So he became the law of it became the law of Mendelian law, or he evolved it out different plants and always conducted several successful experiments with his very keen observation. And of course he didn't have the much sophisticated equipments, but his observation was very wonderful. So then he was very accurate and incisive analysis of the reasons, especially the dominant alleles and the recessive allele. He assessed it in a very fashionable manner, and his knowledge of mathematics was a definite assist for the interpretation. So then another thing, he was such a successful because the plants has got very short life and within a very short span of the time, he could get the results very fastly. Then classified all the plants of a population on the basis of the contrasting characters under his study. He studied thousands of the plants, which he classified them based on the phenotypic characters, contrasting characters especially. Then neglection of Mendel's finding. Later, a lot of research went on, and using mathematical principles was something new and unacceptable to the biologists, especially calculation of the mathematics. So, or predictions, or assumptions, or sometimes the imaginations, which was not accepted by the biologists because they were realistic in nature. They didn't like the imagination, and at the same time, they were very much fond of the truthfulness. But they studied the contrasting fates of the characters exhibiting discontinuous variation, unlike his contemporaries like Darwin, then Galton, and others. So, the phenomenon of fertilization and the behavior of the chromosomes during the cell divisions were not known at that time. During the Mendel's time, the phenomenon of fertilization, especially then the behavior of the chromosomes, especially the changes in the chromosomal pattern, then the chromosomal number, and the different genetic genes, etc., were not known, but no further publishing of his work was there, and he proposed some theory and ended up his life. But it was a great foundation for the future research, even though it is the very basic finding, but his findings were the great foundation, laid a great foundation for the future development of genetic traits. So, the studies of Mendel appeared in establish the consistency of characters of a species generation after generation. So, which was contrary to the theory of evolution at that time, not now. At that time, the other people did not accept it because he appeared to establish the consistency of characters of species generation after generation, which was contrary to the theory of evolution at that time. So Mendel failed to demonstrate the validity of his conclusions in other species of the plants. There are 100 species of the plants, and he only studied the plants with the dominant characters or easily identifiable contrasting characters. So he didn't find it. Whereas the Mendel corresponded exclusively with his contemporary, the noted botanist that's called as he's called as the Nageli. So the botanists they did lot of experiments on different plants and found the difference in the genetic traits. Then biological significance of the Mendel's law. So the biology is totally a different, which will never go on the assumptions and presumptions. But it showed the pattern of inheritance of the character from parent to the offspring. Then the law of independent assortment has laid the foundation of inheritance, which claimed that different genes assorted themselves independently of one another during the gamete formation itself, and the had established the relationship between the dominant and recessive gene type. So, further, it also provided the biological science, the reason for variation in offspring transferred from the parents to the offspring in the form of the gene. That's a, at that time the gene etc. were not properly characterized, so they estimate it as a factor present in chromosomes of the gamete. So the law of segregation, according to this law, when an individual of a species mate, the copy of the genes are separated and assorted so that each gamete receive only one copy. And by the discovery of the meiosis or the cell division, this notion was further confirmed in the biology. Then the this is the basic theory that the Mendel has arrived and genetics has moved far ahead than that of the Rotadia that had been laid down by the Mendel. Now it is an era that moved very far away, and now the new breeds have been evolved, and new traits have been evolved, and many more new things have been evolved. However, the contribution of the Mendel can never be ignored, but still the science owes its existence to the still science owes its existence to the discovery and the ideas that originally been laid down by the Mendel. The same thing is still existing, and it's an evergreen subject. And including the modern geneticists also, they refer the Mendel's law. And in view of this thing, we are uh, stopping. And thanks a lot for being with me. Thanks.